Well, let's start. First, I want to remind you that we're coming close to the end of the semester. And here's what remains on the schedule. Today, we'll talk about molecular genetics. On Wednesday, we'll talk about climate change. And then next Monday, a week from today, is test 4. And test 4 will cover everything that hasn't been covered since test 3. So that would include there, I think I got it. Okay. So test 4 will cover photosynthesis, respiration, evolution, molecular genetics, and there'll probably be a little bit on climate change, but we actually haven't even covered that yet. So we'll cover that on Wednesday, and then something about climate change will be on the test on Monday, the following Monday.
Professor, for the test, is that going to be a 2 time attempt or only 1 time attempt?
2.
Okay.
Any other questions?
Professor,
Yeah.
I had a quick question. So I know like we've been changing the test around and stuff like that, and you've been talking about different test scores. So are you going to be grading on a curve, or is it still the grading that's in the syllabus?
I don't know yet. We'll see how it looks at the end of the semester. My guess is I'll grade on a curve.
Okay, thank you.
I have a question. I feel like this is a dumb question, but what exactly does grading on a curve mean?
It means instead of following a standard set of cutoffs, you grade so that the scores are relative. Each score is relative to the rest of the class.
Okay, thank you.
So if the highest score is 90, then instead of saying 90 to 100 is an A, it would be something below 90 would be an A.
Okay, thank you.
Okay, any other questions? And then to continue on with the schedule, test 4 is a week from today, and then on the 26th, the cystic fibrosis projects will be due. The entire class period on the 26th will be devoted to them. And then a week after that is June 2, and that's the final exam. Okay, the next thing I'd like to do is go over quiz 8. So I'll share my screen. You should be able to see quiz 8, do you?
Yes.
Okay. I'll try to make it as big as I can. Okay, the first question, anaerobic respiration leading to ethanol production commonly occurs in what biological entities? What I had in mind there was yeast, but I would accept bacteria or microorganisms. Alternatively, if anaerobic respiration leads to lactic acid production, it could result in sore muscles. And the reference for this question is these two illustrations of glycolysis from glucose to ethanol, which happens in microbes, for example, yeast. And then we had this slide on lactic acid accumulation in overly tired muscles, which causes a burning sensation. So that's where the sore muscle answer came from. Number 2, choose the correct sequence of steps in glycolysis and oxidative phosphorylation. So I gave you a number of possible sequences to choose from. And the correct one is shown, you can see it clearly on this illustration. So starting with glucose here, the next step is pyruvate, which is then converted to acetyl-CoA, the first step in the Krebs cycle. So the Krebs cycle is next. That produces reducing power for the electron transport chain, the next step, which leads to chemiosmosis, and that makes ATP. So that's the correct sequence in answer to that question. The next one, chloroplast light reaction, starts with the splitting of water. And that's shown in this slide with the electron flow and light reactions. A photon causes water to split into molecular oxygen and protons, and that excites electrons. So the first blank is water. The electrons that are generated flow through photosystem 1, shown here, and photosystem 2, and then photosystem 1, and end up in the chloroplast dark reaction, providing energy for the Calvin cycle to fix the gas carbon dioxide into solid sugar. And that is the main point of this slide on the Calvin cycle. This enzyme Rubisco fixes the gas carbon dioxide into a solid compound, usually glucose. And the glucose then, the output of the Calvin cycle is glucose and other organic compounds. Number 4, under anaerobic conditions, what happens to the Krebs cycle? Well, it would stop. And I refer there back to this diagram, which shows that the final electron acceptor of aerobic respiration is oxygen. Oxygen accepts electrons at the end of the electron transport chain. So if there is no oxygen, there's no receptor, electron transport stops, and that backs up the entire system, all the way back, including the Krebs cycle, all the way back to pyruvate. And it's pyruvate, as you recall, that gets, under anaerobic conditions, glucose just goes to pyruvate and then to either ethanol or lactic acid. But the Krebs cycle and beyond is not involved.
Professor, can you go back to the first slide? Because this one I did get wrong.
Which slide are you talking about? This
The
one?
very first one with anaerobic
Okay.
conditions. You see at the very top where it's got the 2 ATP.
Uh-huh.
Because what the answer I put is that since in anaerobic conditions there is no oxygen, so it would produce less ATP instead of stopping. And I was going from the Krebs cycle versus the Calvin cycle, I guess. You know, where it produces the 38.
Okay, so which question are you talking about then? Number
The
one.
one that we just went over with how it would stop.
Oh, under anaerobic conditions, what happens to the Krebs cycle? And
The answer I put down is, since in anaerobic conditions there is no oxygen, it would produce less ATP.
well, that's true, but it doesn't address the question. The question says, what happens to the Krebs cycle?
Oh, I got it. So I didn't read the question correctly.
Yeah, be careful when you read the questions. Make sure you understand each of the words.
Gotcha. My bad.
Okay, then number 5. Acetyl-CoA is a critical part of oxidative phosphorylation because it is the first step of the Krebs cycle. If you were starving, acetyl-CoA would be generated from what? And the answer is from the oxidation of fatty acids. Again, if we go back to that same figure, it shows you the answer. So if you're starving, you're not getting any sugar. And so this part of the pathway would not be operating, but you have energy stored as fat, as triglycerides. And so they would be oxidized to fatty acids, which then can be further oxidized to acetyl-CoA, and that is the first step of the Krebs cycle. So oxidative phosphorylation could still go on, even if you were starving. And then the other part of that question was, if you had just eaten a large bowl of pasta, where would your acetyl-CoA come from? And it would be from pyruvate. Again, so now you don't need fat because you have lots of sugar from starch, and that is converted to pyruvate, and pyruvate to acetyl-CoA. So the answer to that question is, if you had lots of sugar, acetyl-CoA would come from pyruvate. And then finally, the last question, match the terms. So photosystem 1 is absorption of a photon, and that refers back to this slide, where a photon of light is absorbed by first photosystem 2, and then photosystem 1 absorbs another one. Gas to solid conversion, that's this enzyme Rubisco. I already showed you that. Grana are stacks of thylakoids. That's here. These are grana, so stacks of thylakoids. And the next one is stroma. The answer is dark reaction. Again, shown here, the dark reaction, the Calvin cycle, is in the stroma, which is the space between the grana. Abundant chloroplasts are a mesophyll cell. From this picture, have a leaf, have cells of the leaf, and within each mesophyll cell, there are a lot of chloroplasts. That's where most of the photosynthesis is done. And finally, a proton gradient, chemiosmosis. Chemiosmosis is shown on a lot of slides. This is a good example of it. A proton gradient, you have a high concentration of protons on one side of a membrane, low concentration on the other. so that protons can pass passively through the membrane. And when they do, ATP is generated by the enzyme ATP synthase. So that's the quiz. Any more questions about it? Okay, I've put this in Canvas so you can refer to it when you're studying for upcoming tests. I'd like to stop sharing this screen. You see molecular genetics?
Yes.
Okay. I say it's a complement to Mendelian genetics. In this course, we spend a lot of time on Mendelian genetics, and that's important, but I think it needs to be balanced with some consideration of molecular genetics because they are so much a part of our lives at this point in time. So I've chosen topics that reflect revolutionary technologies that have changed biology and consequently had effects worldwide. The topics I've chosen are restriction endonucleases, the polymerase chain reaction, gene therapy, and finally, genetically modified organisms. And I've also, we've gone over CRISPR before, but I'm going to briefly review it again just to remind you what a revolutionary technology it is and the impact that it's having on biology and medicine. Okay, starting with restriction endonucleases. Back before these things were visualized or even imagined, this would be prior to 1980, people who were trying to work or who were working on the mechanisms of action of nucleases, it was such a boring subject that it was hard for them to get money. And yet when these three gentlemen, Daniel Nathans, Ham Smith, and Werner Arber, discovered endonucleases, it had such an impact on biology that they got the Nobel Prize for it. So I want to put this in context by comparing nucleases with the other polymerases, depolymerases. So if we start with food, food is composed primarily of carbohydrates, fats, proteins, and nucleic acids. There are enzymes to degrade each of these polymers. So we have carbohydrases, degrade carbohydrates, lipases degrade fats, proteases, proteins, and nucleases, nucleic acids. Now, the first three of these types I am calling non-specific. And the nucleases can be highly specific, and that's what's significant here. So let me first tell you what I mean by non-specific. Here on the upper right, again, I'm reiterating, proteases convert proteins to amino acids. Amylases or carbohydrases can convert carbohydrates to small molecules, in this case maltose. Nucleases convert nucleic acids to nucleosides, and lipases digest fats. Now we're going to focus on the nucleases. But first, I'd like to illustrate the non-specificity of these other enzymes. So if we look at proteases, here's a protein, these yellow balls. Proteins are degraded to individual amino acids by 3 different types of enzymes. They're carboxypeptidases, which chew amino acids off of the carboxy terminus of a protein. The aminopeptidases, which do the same thing at the other end. Then the endopeptidases, which make cuts in the middle of a protein. But in all cases, the protein ends up as individual amino acids, and there's no specificity here. Lipases, similarly, you start with a fat and you end up with free fatty acids. No specificity in the target sites for digestion. Carbohydrases, an example here is cellulose from plants. So what kind of enzymes degrade cellulose? Well, exoglucanases start at the non-reducing end and chew off sugars. Exoglucanases can also start at the other end, at the reducing end, and do the same thing, whereas endoglucanases cut polymers in somewhere in the middle. And in any event, you end up with monomers, glucose, from cellulose. So again, there is no specificity here. The enzymes just indiscriminately cut at their active sites. So now I want to contrast the non-specificity with the type of specificity that endonucleases can have. So prior to the 1970s, and I mentioned the guys that won the Nobel Prize were working, sort of wandering in the wilderness, trying to study specificity in nucleases when no one thought there was any. They were thought to digest nucleic acids indiscriminately. And most people couldn't imagine that some nucleases are site-specific. However, there are non-specific ones. So endonucleases randomly hydrolyze phosphodiester bonds, like you see here in the lower left. Endonuclease can cut in the middle of a chain. Exonucleases can nibble off the ends, and these are non-specific. Whereas the last one here, restriction endonucleases, have cut only at certain highly specific sites. Now, where do these enzymes come from? Bacteria, only bacteria. Eukaryotes do not have restriction enzymes. In bacteria, the purpose of a restriction endonuclease is to recognize virus nucleic acid and chop it up. So if a bacterial cell is attacked by a virus, injects its nucleic acid, the restriction endonucleases of the bacteria have sites in the virus DNA and cut at those sites, thus destroying that DNA. The bacteria have methylases by which they hide their own restriction enzyme sites so that they don't digest themselves. And you see in this colored diagram here, the 2 types of cut that a restriction endonuclease can make. In one case, it results in overhangs. It's a staggered cut, and it results in an overhang, which is called a sticky end. And this becomes very important in gene cloning, which I'll mention in a minute. The other type of cut is called blunt end. So instead of an overhang, it's just a straight cut both strands of DNA at the same point. Among the endonucleases, there are various types. They're all site-specific, but in quite different ways. They're called different types, type 1 through type 5. Type 1 enzymes have a specific site where they cut, but they cut about a kilobase away from the site that they recognize. So this is of very little value in terms of molecular manipulation. I've got type 2 in red because it's the type 2 restriction enzymes that are most useful because of the way they act. Their recognition sites are palindromes of 4 to 8 nucleotides in length. And significantly, they cut at the same site that they recognize. In contrast, say to type 1, it cuts a long way away from its recognition site. Restriction or type 2 always cut at the site they recognize. And they yield either a blunt end or a staggered end, sticky. And there are a lot of type 2 enzymes known, roughly 3,500. Type 3 recognize separate non-palindromic inversely oriented sequences, and they don't cut at the recognition site. So they're of little value to molecular biologists. Type 4 enzymes cut methylated sites. So as I said over here, DNA can methylate their own restriction sites, so they're not cut by their own enzymes, but type 4 enzymes will cut them. And type 5 I've highlighted because type 5 enzymes use guide RNAs to target specific non-palindromic sequences found in invading organisms. So they cut DNAs of variable length. And a good example of a type 5 is CRISPR-Cas9. CRISPR-Cas9 is a non-specific endonuclease, but it cuts at a site that it's guided to by the guide RNA. And I'll review this just briefly a little later when we talk about CRISPR. And finally, DNA ligase is not a restriction endonuclease, but you always see it along with restriction endonucleases because it's needed to join the ends of DNA. It's an end joining enzyme. You may remember when we talked about DNA replication, the last step is DNA ligation. There's always a gap left when two DNA ends are joined. And so that gap has to be filled. That's what DNA ligase does. So to review, the nucleases can be nonspecific, just like with the other types of depolymerases, or they can be specific. And they can be specific in a number of different ways. And these ways are numbered type 1 through type 5. And it's the type 2 restriction enzymes that we're going to focus on now. They're the ones that are useful in molecular biology. Here in the upper left are some examples of type 2 enzymes. The recognition site can be anywhere from 4 to 8 nucleotides long. The shorter it is, the more frequently it occurs in a genome. So for example, if the recognition site is 4 nucleotides long, that occurs about every 256 base pairs. So there are a lot of them. A 4 cutter chops up the DNA in very small bits. On the other hand, an 8 cutter has an 8 nucleotide recognition site, cuts every 65,000 base pairs. So very rarely. And so if you want very large pieces of a genome, you use an 8 cutter. And below that is a list of some examples of restriction enzymes, where they come from, and what their recognition sequence is. So EcoR1 comes from Escherichia coli. And here you see its recognition site, GAA-TTC, on one strand. And on the other strand, it's just the opposite, GAA-TTC. That's called a palindrome. And all of the recognition sites for type 2 enzymes are palindromes. And that's a little bit difficult to comprehend at first. So let me give you another example, a couple examples of palindromes. A man, a plan, a canal, Panama. I'll read that backward, starting from the right and going left.
It's the same thing.
You see how it works? It's the same thing, but backward. And the next one is also like that. Able was I ere I saw Elba. Again, if you want to read that backward, it says exactly the same thing. So those are palindromes, except in the case of DNA, they're nucleotides instead of alphabetic letters. Most of the enzymes listed here make staggered cuts. One doesn't, the last one, PVU2, makes a blunt end cut. Now, I'm going to talk about cloning. And for cloning, you need a vector. And vectors are always plasmids. So let's talk about what is a plasmid. Here's a bacterial cell. It has a nucleoid. Its genome is just dispersed DNA strand. Completely independent of that are plasmids. Most bacterial cells have plasmids. So what are they? They're molecules of DNA. They replicate independently of the chromosome. They're usually circular, but not always. Some of them are linear. They're found mostly in bacteria. They're also found in eukaryotes. And for DNA cloning, they are essential. Now, on the right, you see a generic plasmid, cloning vector, which has a site, usually called a polylinker, which has a whole series of restriction enzyme sites there for the convenience of the cloner, has a replication of origin, an antibiotic resistance gene, and a selectable marker. So that's what plasmids are. And they're essential for cloning. This is another description of them. Their functions in bacteria, their natural functions, some of them encode toxins, and others encode genes for resistance to antibiotics. Now let's look at the right. This is how you clone a piece of DNA. So start with this red circle, plasmid, which has a cleavage site. And it turns out the site used here is for EcoR1. If you look over here at EcoR1, after cutting the recognition site, the overhang is AATT on one strand, and TTAA on the other strand. So you see that after the plasmid is cut, it has these so-called sticky ends, the overhangs, which are TTAA and AATT. Now the DNA that you want to clone, it's called here foreign DNA. So let's just say it's DNA from the genome of some bacterium. You cut it with the same enzyme. So you cut it also with EcoR1, so that now the fragments that you get have sticky ends, overhangs of AATT on one strand and TTAA on the other strand. So these sticky ends will stick to the sticky ends on the plasmid simply by hydrogen bonding. Remember, A's always bond to T's, T to A, T to A, T to A. And on the other end of the fragment, same thing. So now the foreign DNA has become a part of the plasmid with one thing left to do, and that is seal these gaps at the end. And that's where DNA ligase comes in. So then you have a complete plasmid molecule, but now instead of just being a plasmid, it's a plasmid with a piece of somebody else's DNA. So is that clear? How the steps in cloning and why it works, why it's, once you know how to do it, it's relatively easy to do. Nomenclature, naming restriction endonucleases. Where do these exotic sounding names come from? Well, the rules, and these are followed rigorously, the three letters, like ECO, three letters that correspond to the scientific name of the microorganism that makes the enzyme. So in the case of E. coli, it's E from Escherichia and CO from coli, E. co. And then that indicates the bacterial source of the enzyme. The R1 designate which enzyme it is. So E. coli produces a lot of different restriction enzymes. There's an EcoR2, an EcoR5, and several others. Similarly, with Haemophilus influenzae, it's named HIN. H from the Haemophilus, IN from the influenzae. And then the D3 specifies a particular restriction endonuclease produced by Haemophilus influenzae. So there's always a unique identifying code like R1, H1, D3, and so on. So is that clear how restriction enzymes are named? Once you know what the rules are, it's fairly easy to understand the names. And now I'd like to talk about the usefulness, how you might use cloning to make a genomic library. So supposing you want to clone all the DNA fragments from a bacterium or from a fungus or from a mouse or a human. The first point you have to consider is that most genomes do not code for proteins. So what you want in a library is only the protein coding sequences. And that's usually less than 5% of the total genome. So how do you get just the ones you want? Well, you do it by, instead of starting with the isolation of DNA from your organism, you isolate messenger RNA. Why mRNA? It's because mRNA accurately reflects the protein coding genes. If a gene does not code for protein, it does not make a message. Then you remember reverse transcriptase. We talked about this earlier. DNA is transcribed into RNA and RNA into protein, but the first step can be reversed with this enzyme reverse transcriptase. So RNA can be made back into DNA. And why is that important here? Because you can't clone RNA in a DNA vector. The vectors are always DNA. So you reverse transcribe the messages into what's called complementary DNA, cDNA, with reverse transcriptase, and then you treat that as your library. You cut it with a restriction enzyme, and you cut a vector with the same restriction enzyme. So again, this is just straightforward cloning like we saw on the previous slide. And so we end up with a series of vectors, of plasmids. Each one is a chimera, that is, each one has an insert of a piece of the target DNA, and each one is different. Each plasmid is different. So if these are transformed into E. coli, one plasmid per cell, E. coli will faithfully produce them. And you can have a clone of 100,000 or 1,000,000, however many you want, different clones, independent clones of your genome in E. coli cells. And these can be single-celled and isolated into test tubes, and that's the library of DNA, called a cDNA library. And the steps that I just have gone through, I have written out here on the left. Any question about how you clone, how you make a library? Okay, some of the uses for restriction enzymes. To make recombinant DNA is one of the main reasons, make genomic libraries, restriction maps, DNA fingerprints, and how are all these things done? The technology is fairly straightforward. So if you start, for example, with a restriction map of a virus, bacteriophage lambda. So here you have at the top of the center diagram, the genome of lambda from 0 base pairs to 48,000. Below that, if you digest that with BAM-H1, you end up with 6 fragments. 1, 2, 3, 4, 5, 6, and they're each a different size. Now, if you cut the same thing with a different enzyme, EcoR1, again, you end up with 6 fragments, but they're all different sizes, different from the BAM-H1 digest because the sites are different. BAM cuts at a different site than ECO. So if you do that and you want to visualize or even isolate these little pieces, you can do it with a process called polyacrylamide gel electrophoresis, called PAGE. And this is done by making a gel. You melt the polyacrylamide and pour it into a mold with little holes at one end, little wells. You load your sample. So you take the digest, put it in a well, and then you put that in an apparatus that's hooked up to current. Now you remember DNA is an acid. It's negatively charged. So in an electric field, it will migrate toward the positive end. All the DNA fragments, as soon as you turn on the power, they start to move through the gel. However, they do it at different rates depending on their size. So long pieces go slowly, short pieces go fast, as you can see. Here at step 5, this is the gel after it's stained, or in the center here, you can see an actual picture of a gel. At the top, you can see the little slots where the samples were loaded. And you can see then that the current was turned on and the fragments moved through the gel at different rates. So the Eco R1 digest made, it should have made 6 bands, 1, 2, 3, 4, 5, 6. Here's 1, 2, this is a double band, 3, 4, 5, 6. And similarly, the BAM digest here should have made 6 fragments. Again, 1, 2, 3, 4, 5, 6. But the BAM fragments are all different size from the ECO fragments. Now, this is how they're visualized. And if you were actually interested, if a gene that you wanted was on one of them, say this 7.4 kb fragment, you could actually cut this out of a gel and clone it. Now, this technology is important for a lot of different reasons, but one in particular is forensics. Now, this is a DNA fingerprinting of a rape investigation. So the DNA was taken from the victim, and then there were 3 suspects, A, B, and C. And then here is DNA taken from the site of the crime. So you can see that the DNA at the site of the crime had these 2 distinctive bands, and they match precisely with suspect A's DNA. Clearly different from the victim, clearly different from the other 2 suspects. So A is the culprit. Any question about how this works? This has real, this technology has so many practical applications, and this is just one of them. Now I want to switch topics to the polymerase chain reaction. What is it? It's the synthesis of large quantities of a specific DNA segment, starting with only a little bit. And that little bit could be as little as 1 molecule. Now I'll go through this step by step with you, but first I would like to play this video.
Hi, this is 2 Minute Classroom, and today we're talking about polymerase chain reaction, or PCR as it's more commonly known. First, let's talk about what PCR is and how it works, and then I'll give some examples of what PCR is used for. PCR is a technique used to amplify or copy small specific segments of DNA. For PCR to be successful, you need the DNA sample you want copied, DNA primers to identify the specific sequence of DNA you want to amplify, lots of raw nucleotides to form the DNA copies, and a heat-resistant DNA polymerase to make the new copies. This all goes into a special, stable solution. Once all the ingredients are in, you run the solution through many temperature cycles until you reach the amount of DNA copies needed. There are 3 steps to this cycle. Step 1 is denaturation. The solution is heated, allowing complementary DNA strands to separate so that all the DNA in solution is single-stranded. Step 2 is annealing. The solution is cooled so that the DNA primers can bind to the specific region of DNA you are interested in. Step 3 is extension. The temperature is raised so that the DNA polymerase can bind and perform the necessary operation of synthesizing new DNA strands. This cycle is repeated about 30 times, takes about 3 hours, and creates over 1 billion copies.
I don't think
So
we
what
can
is
see the
PCR actually
video.
used for? PCR has many research-based and practical applications. It's used for DNA mapping, like in the Human Genome Project. It's used to check sections of DNA for genetic testing to determine potential disorders. Companies like 23andMe and Ancestry.com use PCR to run personal DNA tests. And my favorite application for PCR is in forensics. Whenever a crime is committed, the perpetrator almost always leaves their DNA at the crime scene. This could be hair, semen, skin, or even blood, for example. Investigators search for this evidence and, thanks to PCR, can amplify the DNA and compare it to the DNA of suspects. This genetic information is then put into the FBI's.
Professor, I don't think I see the video as well.
It's even better.
You don't see the
Investigators
video?
have been able to
Yeah, no,
No.
we can't see it.
crimes committed long before CODIS was put together. Thanks for stopping by. This is 2 Minute.
Do you see it now?
Yes.
Yes, I can see now.
Genet classroom, and today we're talking about polymerase chain reaction, or PCR as it's more commonly known. First, let's talk about what PCR is and how it works, and then I'll give some examples of what PCR is used for. PCR is a technique used to amplify or copy small specific segments of DNA. For PCR to be successful, you need the DNA sample you want copied, DNA primers to identify the specific sequence of DNA you want to amplify, lots of raw nucleotides to form the DNA copies, and a heat-resistant DNA polymerase to make the new copies. This all goes into a special, stable solution. Once all the ingredients are in, you run the solution through many temperature cycles until you reach the amount of DNA copies needed. There are 3 steps to this cycle. Step 1 is denaturation. The solution is heated, allowing complementary DNA strands to separate so that all the DNA in solution is single-stranded. Step 2 is annealing. The solution is cooled so that the DNA primers can bind to the specific region of DNA you are interested in. Step 3 is extension. The temperature is raised so that the DNA polymerase can bind and perform the necessary operation of synthesizing new DNA strands. This cycle is repeated about 30 times, takes about 3 hours, and creates over 1 billion copies. So what is PCR actually used for? PCR has many research-based and practical applications. It's used for DNA mapping, like in the Human Genome Project. It's used to check sections of DNA for genetic testing to determine potential disorders. Companies like 23andMe and Ancestry.com use PCR to run personal DNA tests. And my favorite application for PCR is in forensics. Whenever a crime is committed, the perpetrator almost always leaves their DNA at the crime scene. This could be hair, semen, skin, or even blood, for example. Investigators search for this evidence and, thanks to PCR, can amplify the DNA and compare it to the DNA of suspects. This genetic information is then put into the FBI's DNA database, CODIS. But it gets even better. Investigators have been able to take DNA evidence from crimes committed long before CODIS was put together and run it through the current system. This has solved many cold cases and also exonerated prisoners who had been wrongfully accused, even some that were on death row. Needless to say, PCR is one of the most important scientific advances ever. And now you know why. If you have additional questions, I'll do.
Now are you back with me? Polymerase chain reaction. Can you see that?
Yes.
Yeah, we can see
Yes.
it.
Okay. All right, so this is where we left off before the video. So what is it? As the video said, it's a way to synthesize large quantities of a specific DNA segment. And the really important part of it is that you can start with very small amounts of starting material, even if it's only one molecule. So I'd like to review what the video just showed you, how PCR works. What are the components of PCR? Well, first you have to have a target DNA template. So on the right here, you see a strand of genomic DNA, and this one little blue region is the target. You want to make a lot of that. So you make up a reaction mixture that has DNA polymerase. That's here, Taq polymerase. It's from a bacterium that was taken from a hot spring, Thermus aquaticus. And this, I've actually mentioned this enzyme before because it has this unusual characteristic that it doesn't denature even at temperatures near boiling. So it can tolerate that, and of course it has to in this bacterium that came from a hot spring. So not only does it not denature at high temperature, its actual catalytic optimum is at 70 degrees centigrade, which is still pretty hot. Okay, so you have to have Taq polymerase. You have to have primers that correspond to the ends of your target. So here are the primers in your reaction mixture. And you have to have the DNA triphosphates. So the A's and T's and G's and C's that are needed to make DNA. So they're part of the reaction mixture. And then you have to have a buffer. So you start out, take this mixture that's got, now it's got your target and all the components, and you heat it to maybe 95 degrees. And when that happens, all the hydrogen bonds in the target DNA are broken and the two strands come apart. Then you cool down to about 50 or 60 degrees. And it takes just a few seconds at the high temperature and another few seconds at a lower temperature so that the primers, these guys, can anneal to their target sequences at the ends of the target DNA. Then you raise the temperature up to where Taq polymerase, the DNA polymerase, is optimally active, and that would be around 70 to 75 degrees. Hold it here for a couple of minutes and it's done. So now you've gone from one molecule of target to two. You now have two molecules. And now you do it again. And this is all automated. The temperature cycles are completely automated now. And so cycle 2, you again heat it up to denature, allow it to the primers to anneal, and raise the temperature to 70 degrees for Taq polymerase to act. And now at the end of cycle 2, you've got 4 molecules. Do the same thing, cycle 3, and now you have 8 molecules, starting with just 1. After cycle 5, you have 32 copies. After cycle 15, you have 32,000 copies. And after cycle 25, you have 33 million copies. So you can see why it's called an amplification. It amplifies the DNA from whatever you started with to as many copies as you want. If you want more than 33 million, just keep the cycles going. So is that clear? PCR is conceptually very simple, but it's an extremely powerful technique, and it has had as much of a revolutionary effect on biology as CRISPR or endonucleases. I'd like to tell you a little bit about the history of PCR. It's not really the way science is often done. And it may have been sort of a once ever opportunity for this technique to be developed and discovered. But prior to PCR in the 1980s, the only way to get DNA was to isolate it from an organism. And that usually meant cloning it in E. coli, and then you grind up the E. coli cells, extract the DNA, and purify it. But then, as I said, in 1969, this man, Thomas Brock, isolated this new species of bacterium from a hot spring. And from that came Taq polymerase, this enzyme that can withstand boiling temperatures, and its optimum catalytic activity is at 70 degrees. Well, in 1979, a new biotech company in San Francisco hired a man named Kary Mullis for a specific job, which he never did. Instead, he was interested in other things, and among his interests developed in 1986, he presented a talk at a symposium in which he announced this polymerase chain reaction. Then at about the same time, he left CETUS and went on to other things. In 1993, he got the Nobel Prize in chemistry for developing PCR. And an interesting side note here, when you get the Nobel Prize, you have to go to Stockholm and make a speech. In his speech, he acknowledged that he had worked very hard, almost continuously, because he was very excited about PCR. The night he proved that it would work, his girlfriend left him because he had virtually ignored her while he was working on PCR. And a final note, financially, when Mullis left CETUS, they gave him a bonus of $10,000 for this work he had done. CETUS turned around and sold the technology that Mullis developed to Roche, another pharmaceutical company, for $330 million.
I have a question.
Yeah.
So is the Thermus aquatis from the Yellowstone National Park like the only Taq polymerase used in this, or is there other forms now?
There are others from other heat-tolerant bacteria, but TAC1, which is the name of this one, works best. So if you order reagents from a lab supply company and you order DNA polymerase, you'll always get TAC1.
Oh, okay.
Professor, I have a question.
Uh-huh.
This is here. I was just wondering the difference between PCR and CRISPR. Like, what is the main difference?
The main difference is that PCR amplifies DNA, CRISPR makes mutations. So PCR does not cause mutations in DNA. It just makes a lot of it in a short time.
Okay. Thank you.
Mm-hmm. Anything else? I have pictures here of Kary Mullis. He was a real character. I'll tell you a little more about him in a minute, but applications of PCR, it's used all the time in a lot of different places. Whenever a large amount of DNA is needed, and especially if the amount of target DNA is very small. So for analysis of clinical specimens, for example, the coronavirus and COVID-19. In fact, PCR is the basis for the standard test that CDC uses. If you go to some place and get tested for coronavirus, your sample is tested with PCR. You can use it to identify mutations. And you already heard about forensics. As little as a single molecule from a crime scene can be amplified, and it can be proven who that came from. Now, we talked about Neanderthals, ancient DNA. And if you recall, when we were talking about that, I showed you this slide. I said, how do we know so much about Neanderthals and humans? One man, Svante Pääbo, who in 2010 sequenced the genomes of 3 Neanderthals. How did Pääbo do it? PCR. He went into this cool, dry cave, got some Neanderthal bones, still had, not intact, but still had fragments of DNA left in them. So he used PCR to generate the entire coding sequence of Neanderthal people. So that's a very powerful use, getting DNA from fossils, mummies, sex determination, and so on. For virtually all molecular genetics research uses PCR in one way or another. So the key points in the development of PCR was, first of all, the discovery of Taq polymerase, and secondly, the hiring of Kary Mullis by Cetus, who, instead of doing what Cetus wanted him to, went off on his own and ended up developing PCR. If he hadn't done this, question is, would it ever have been done? And would we have never gotten the opportunity to use this technology? Now just, again, we're running out of time, but I take a couple of minutes to tell you a little about Kary Mullis. He was a character and not a typical scientist. There is no such thing as a typical scientist, but he was really out on the edge. He was an oddball, an iconoclast. PCR was his only scientific contribution. He didn't do anything before that. And after he did it, got the Nobel Prize, he didn't do anything else. He left science. He had a PhD in biochemistry. For a while, he wrote fiction, and he was a manager of a bakery. And one of the odd things he did was, after he developed PCR, he used it to amplify the DNA from famous dead people like Elvis Presley and Marilyn Monroe. And he embedded the DNA in metals and sold it as jewelry. He was a skeptic and a conspiracy theorist. He didn't believe in climate change. He didn't think there was an ozone hole in the atmosphere. He didn't think that HIV caused AIDS. And he was an advocate of hallucinogenic drugs. In fact, he claimed that LSD gave him the insight to develop PCR. He died a couple of years ago, 2019, at age 74. And these cartoons just indicate some of the outrageous things that Kary Mullis actually said.
Professor?
Yeah.
How many times do they run PCR? Like, nothing's perfect, so how do they account for like mistakes? Do they run it multiple times, or is there like handwritten stuff that they do to back up PCR, or how does that work?
mean, how to determine the fidelity of it?
Yeah, like sometimes like mistakes happen. So do they run it multiple times or do they just once and then what PCR says is what it is?
It's, yeah, the fidelity of PCR is pretty high, but you're right, mistakes can be made, but they would be discovered fairly quickly because you run the products of a PCR reaction on a gel, and you can predict the sizes of the fragments you should get. If you don't get those, and you know something is wrong. And yes, you're right, you then would throw out the entire reaction mixture and start over.
Okay, got
But
it.
the fidelity of it is so high that for all intents and purposes, if you run a PCR reaction, what you get is the correct amplification. Okay, gene therapy, and we're out of time. So this is the last thing that I'll talk about today, and we'll leave the rest for next time. Basically, two ways proposed, and some of these are being used. If you have a mutated gene and you want to correct it, you can either replace it with the wild type sequence, or you can add the wild type sequence to it. And that's illustrated here, gene addition and gene repair. So say you've got a mutated gene here, and you've cloned it in a stem cell. You can put wild type DNA, the wild type of this gene, in a viral vector and infect the cell with it and hope that the normal gene product outcompetes the mutant gene product. Now, another way, and probably better way to do it, is to replace the mutation, called gene repair. So again, you have the wild type sequence in a viral vector. So the vector then inserts that wild type sequence into the cell with the mutant gene. And then you initiate a recombination event, a double crossover, whereby the homologous sequences on wild type and mutant recombine, and the wild type sequence replaces the mutant sequence. So now you've gotten rid of the mutation, you have nothing but wild type. So those are a couple of ways that people are thinking and actually using recombinant DNA to address disease issues. So I've used up all the time now, and I'll stop at that point. Any quick questions before we go?
Professor, I have a quick question. We have a test on Wednesday, right? I mean,
No.
a quiz.
No, there's no quiz on Wednesday.
Oh, okay. Because I see on the announcement and it said that there's a quiz on Wednesday this week.
Well, I'll have to look at that. I don't think so, but I'll look at it again. You mean the announcement that I just sent that had the schedule for the rest of the semester?
Yes, it has the past quiz, the previous quiz, and the quiz that I was asked you on Wednesday, and then the quiz on next week.
Well, test 4 is a week from today. Anyway, I will check that announcement, and if there is a mistake on it, I'll send another announcement indicating that it was a mistake.
Okay, so no quiz.
So-
There's no quiz on Wednesday.
Okay.
Next Monday, we won't be having class. We'll be just taking our test.
Yes, that's right. So no regular class on Monday. You'll have all day to take the test. The test will be 75 minutes as usual, a regular class period.
Are we gonna have 2 attempts on this one again?
Yeah,
Thank you.
Anything else?
Yeah, I just had one question. The PCR technology, is that what led to them coming up with CRISPR technology?
No, no. PCR was developed completely independently of CRISPR.
So it's two totally different technologies.
Yeah. CRISPR was the brainchild of this one crazy guy, Gary Mullis, and he developed it, and it just turned into a blockbuster invention. Whereas CRISPR was discovered by studying the bacterial immune system. It's another way that bacteria have of destroying viruses. Endonucleases is one way, and CRISPR is another way. The bacteria, it's like a memory. CRISPR is a memory system. Once a virus invades, the bacteria make copies of little segments. And so if the virus comes back again, they're ready for it.
Oh, cool, thank you.
Professor, I checked and it was the announcement under the week, I think week no quiz 8 announcement.
Quiz 8?
Yeah, quiz 8 announcement, and you
We've
put the
already,
rest,
yeah, we've already had quiz 8.
yeah, under that announcement you put the rest of the quiz, the schedule. That I think was confusing.
Okay, all right, I'll check it and I'll try to correct the confusion.
I had a question about my about a grade.
Yeah.
On the extra credit opportunity thing, if I like lower it, it lowers my grade. So would that make it an extra credit? Because
Yeah. Yes, it's a good point. The extra credit was given to you as an assignment.
Mm-hmm.
So it takes a spot on the semester schedule. So you'll get whatever credit, whatever number of answers you got right for that assignment. And then that number again will be added at the end of the semester. So that's where the extra credit comes in.
Okay.
So when it's added at the end of the semester, it will not occupy a spot as an assignment.
I see the announcement. It was posted on May 9th. It just says that we have it's just in that announcement from May 9th. It says we have a quiz on the 19th, quiz 9, covering evolution and molecular genetics.
All right, well, that's a mistake then. I'll send an announcement correcting that. Okay, thank you for pointing that out.
Thanks, Professor. Have a good night.
Okay, see you all Wednesday.
Oh, I had one final question, a personal question about grades. I've noticed this and I'd never said anything. I don't know why, but for some reason on the grades, there's 2 quiz 6s that show up. One has a grade and one says missing, but they both say quiz 6.
That I cannot explain. That missing quiz 6 just got into the grade book. I don't know how or why. It doesn't seem to be doing any harm, so I haven't done anything about it. But the legitimate quiz 6 is there.
Okay, cool. Thank you.
Mm-hmm. Okay, see you Wednesday.
Bye.
Thank you, Professor.
Mm-hmm.